think we will wait a few more minutes so that more people can join this session today. So keep online. Okay, great. So I think there are lots of people today in this session. So welcome to QBiotech's scientific masterclass series, where researchers from around the world present their latest results and findings to a global audience. My name is Michael Erklens, and I'm part of the research and development team of QBiotech. So I'm happy to introduce this masterclass series today, which provides a unique platform for scientists to share their work, connect with others in their field, and receive feedback from experts and peers. After the talk today, there will be time for the interactive question and answer session. So during the talk, you will be able to use the chat function you see on the bottom right to comment direct the aspects of the talk, or you can place a question for the question and answer session by changing the chat mode to the Q&A mode. So you can see on the bottom right, there's a little question mark, and then you can place your question. So our speaker today is Dr. Kirill Nadezhdin, and he's a postdoctoral researcher at the Columbia University Irving Medical Center in New York. So welcome, Kirill. So you can turn on your camera.
Hello, everyone.
Hi, Kirill. So nice to have you on board today, and that you will explain us your topic today. So Kirill completed his undergrad studies at the Moscow Institute of Physics and Technology, where he focused on mathematics and physics with applications to biological systems. He earned or obtained his PhD in biophysics at the Lomonosov Moscow State University while researching the amyloid precursor protein associated with Alzheimer's disease. Following that, he joined the Columbia University in the city of New York as a postdoctoral research scientist. There he specialized in studying the structures of ion channels using cryogenic electron microscopy. At the moment, he is a postdoctoral researcher in the Sobolevsky lab and has a quite impressive publication history, as 6 out of 7 recent publications were published in the same volume of Nature Communications this year and at the end of last year. So it's really impressive. And so with this, I would like to finally hand over the word to Dr. Kirill Nadezhdin and his really exciting talk. So maybe you also saw him at the last SMART meeting. Yeah, so feel free to start whenever you are ready.
Yeah, thank you very much for this kind introduction. Let me
You're welcome.
My presentation and okay, let's get started. So hello everyone, my name is Kirill Nadezhdin, and I do cryo-EM in Columbia University in the city of New York. And today I'm going to talk about one of our recent works when we study magnesium channel TRPM7. So First, I want you to introduce the minerals in human body. As you know, we have a lot of cations and minerals in human body, and the most abundant one is obviously calcium. Then it is followed by potassium, sodium, magnesium, iron, zinc. And what is interesting that the distribution of all these minerals throughout the body is not equal. There are more minerals outside the cell than inside the cell. And it is interesting that one of the minerals, magnesium, is very abundant in the cells. And why it happens so? So this is linked to charge density of cations. And as you can see in this table, that there are several minerals, several cations that have very high charge density. I'm not going to talk about iron because it does different stuff. But the most abundant cations are sodium, calcium, potassium, and the charge density is very low. And for magnesium, the charge density is pretty high. It means that it is very reactive. And let's talk about magnesium a little bit more. So our bodies, they don't produce minerals. So we need to eat food to obtain all these minerals. And you can find a lot of magnesium in nuts and seeds, dairy, legumes, leafy greens, dark chocolate, water, but mostly in seawater. You can find a lot of magnesium. And what is interesting about magnesium, that it is the second most abundant cation inside the cells, and it participates in like numerous biochemical reactions, more than 300. Why? Because it has very high charge density. And it participates in energy production, protein synthesis, muscle, nerve function, and it forms complexes with proteins, DNA and RNA. And like, for example, the magnesium is essential for ribosome function. And there are a lot of illnesses that are associated with magnesium deficiency. And most known of them are cardiovascular disease, metabolic syndrome or insulin resistance, chronic fatigue syndrome, that is very crucial nowadays, kidney failure, Alzheimer disease, cancers, and others. So what happens with the magnesium when it enters the body? So it is absorbed in intestine, in colon. So it goes through the enterocytes of colon, through paracellular pathway mostly, and also through the transcellular pathway. Then it goes to blood vessels, and then it is stored mostly inside the cell. So every cell in human body has magnesium, and it is essential mineral. So not all of course, but many biochemical reactions inside the cells, they rely on magnesium. So again, like every cell, neurons, epithelial cells, red blood cells, they all need magnesium to work. And the excess of magnesium is stored in bones and red blood cells. And in our lab, we work on transit receptor potential channels. And this is the tree of these channels in mammalians, in animals. And why they are so interesting? Mostly they are mostly interested because they sense temperature. Not all trip channels sense temperature, but many of them sense temperature from cold temperatures to high temperatures. And it is interesting that some of trip channels, the TRPM7, it is considered to be the master regulator of divalent cations in human body, and it is also considered to be the magnesium channel. So we do cryo-EM and structural biology in our lab, and we wanted to know how this channel works. So for that, we need to express the channel and do structural biology with it. So let's dive a little bit deep into it, how we do this. So we use several cell lines to produce the protein. We use two types of bacteria, and we use insect cells and mammalian HEK cells. So first we produce plasmids in E. coli. Then we use different strain of E. coli to produce backmids. And after that, we use backmids and we transfect SF9 cells, insect cells, to produce virus, baculovirus. Then we harvest this virus and infect HEK cells, and then those cells become infected with the virus, and then they can produce the protein on the cell membrane. So let's talk a little bit more in depth how we express the protein. I'm not going to talk much about how we make plasmids. This is we do this using standard procedures. So let's start with backmids. So we transfect backmids. We transfect SF9 insect cells with backmids, and we grow for about 5 days. And SF9 cells, insect cells, they produce virus. Then we harvest virus, though it is very weak and the concentration is very low. So we use the same virus and we do scaling up. So we use this virus and we infect more SF9 cells, insect cells, to produce more virus. Then we harvest this virus after several days and concentrate it. And after that, we infect HEK mammalian cells with this virus, express our protein for 2 to 3 days. After that, we harvest cells, we pellet them, and store them for future structural studies. And in total, this protein expression takes about 2 weeks from backmid transfection, SF9 transfection, to the final cell pellet. So how we purify the protein? So this is the outline, simplified outline, how usually people purify protein from HEK cells, and it has several steps. First starts with cell lysis. So we lyse the cells with the sonication, but people also do French press for that. Then we centrifuge the cells, and all cell organelles and debris, they are pelleted, and our protein now in the supernatant. Then we use ultracentrifugation and we pellet membranes, because our protein is ion channel, it is membrane protein. So we pellet our membranes. Then we take this pellet and homogenize this with the Downs homogenizer. Then we add detergent to extract protein from the membranes. We add resin to the extracted proteins. So this resin, our protein has specific tag, so it only our protein binds to the resin. Then we use simple affinity chromatography to purify our protein, because only our protein has tag. And we use additional step, size exclusion chromatography, where we additionally purify our protein from aggregates, other proteins. So, and when we have our protein, we do cryo-EM, we make grids. So that's exactly what we do when we started this project. And in the end, we got the beautiful structures of the TRPM7 channel, magnesium channel in detergent at decent, good resolution in closed, so-called apostate, resting state. And we also introduced one mutation to our channel to make this channel constitutively open, always open. So we also got the structure of TRPM7 channel in the open state with the mutation. And there are some summary from this project. So we saw the structure in apostate, resting state. It is good. We also saw the structure in activated open state for the mutant, and it was excellent quality of the structural results. But can we do better? Actually, we have some not very good results. So we obtained the structure of open state for the mutant. So can we do this without mutation? So we also obtained the structure in detergent. Specifically for this project, we used this detergent called GDN. And sometimes it is reviewers and other people in the field, they kind of hesitate whether detergent gives the native state of the protein. So this is like huge question mark. And we also try to open the channel with the agonist, but for some reason, agonist doesn't bind. So we tried, it didn't bind. And on top of that, the protein was unstable and the protein yield was low. So can we do better with this protein? Okay, so what can we do? So we can improve the yield, we can make it more stable, and maybe we can try to do open state without mutation. And that's what we tried to do. So let's go back to the protein purification. So can we do better? So actually we can. So we can try to optimize all these steps because our protein is unstable. So maybe if we can shorten some steps and make it like faster, or just discard some steps, maybe the protein, we can get more protein. So that's exactly what we did. So it turned out that if we just eliminate these two steps, cell lysis, centrifugation, and homogenization, we still have a lot of protein, even more. And so we can start with the extraction. And this is exactly what people do when they use, let's say, smolts. They just use cell pellet, they add smolts, they extract a protein from cell membrane. Yes, we did the same, but we use detergent for that. And all other steps are the same, and the yield is better. Okay, can we increase the stability of the protein? Actually, we can. So we increase the yield. So now it was enough protein to try to reconstitute this protein into nanodisks. So what we do when we reconstitute protein into nanodisk, we take our protein in detergent, we add lipids, and we also add nanodisks. Those are proteins that kind of form belts around the protein and protect transmembrane domain from water. So when we mix everything, we incubate it for about 1 hour. Then, like the magic step happens, we add biobeads. And biobeads, they bind all detergent from the solution, and it forces the nanodisk it forces this nanodisk protein to wrap around our receptor. So we incubate for quite some time. And after that, we remove biobeads and do site exclusion chromatography, where we just get rid of empty nanodisks. We prepare grids using cryo-EM, we solve the structures. How can we assess the quality of our protein? Actually, it's pretty easy. So when you do site exclusion chromatography, what you always expect something like a good narrow peak of your protein. And for the protein in GDN, yes, it was like very narrow, beautiful peak. In nanodisk also, and for the mutant. So even site exclusion chromatography gives you some information about the quality of the protein. In our case, it was good. And then you can also use cryo-EM to assess the quality. So what we do in cryo-EM, we collect thousands of these micrographs. And you can see on these micrographs, there are like some specs. So these are our proteins. So we collect thousands of micrographs. We extract particles from these micrographs. Usually it's count like millions or like hundreds of thousands. We align them when we get these beautiful 2D classes from cryo-EM. And after that, we do reconstruction and got our maps. So this is the beautiful structure of TRPM7 at very good resolution, 2.19, way better than in detergent. And as you can see, the quality is good. This is the micelle. We see a lot of lipids that are bound to our channel. And on top of that, we see some interesting stuff. So we also see specific lipids like cholesterol that is bound to our protein. And this is interesting because we didn't add any cholesterol during protein purification. So whatever is bound to our protein, if we see cholesterol that is bound to our protein, it means that it came from the cells itself. So during all steps of our purification, during the extraction using detergent, during reconstitution into nanodiscs, this cholesterol molecule still stays attached to our protein. So it means that cholesterol is very important for the integrity and the protein structure. So, and on top of that, we improved resolution, but we also got the structure of the open state in nanodiscs with the activator that is called Ultraman. So we couldn't do this in detergent, but in nanodiscs, it was possible. So, and you can see that the density of this activator is pretty nice. But is it open? So we actually, if you compare this channel to the closed state, the pore is wider. So that was a very good sign. We were very happy about our results. So when you do cryo-EM and you do so-called structural pharmacology of membrane proteins using cryo-EM, you always have to be very cautious that basically you add some hydrophobic molecule to your hydrophobic protein and it can just stick to it. And it doesn't mean that it does anything or binds in specific spots. So you have to provide some additional evidence that the molecule that you see in your maps is actually the binding site of this molecule, or not just some unspecific binding. So this is exactly what we do. We mutated all residues around this nitroben binding site. And as you can see, this is the black one line for the wild type, no mutations. And you can see these activation curves of nitroben, of TRPM7 by nitroben, they are shifted. It means that all these residues around nitroben binding site, they sense the presence of nitroben. So this is kind of additional evidence, additional justification. Yes, this is indeed the active binding site. And we also did some experiments with single channel recordings because our protein had tag attached to it. So maybe this tag may influence the function of the channel, but it looks like that without adding nitroben, nothing happens. When we add activator, it does open. So it's kind of additional physiological evidence, yes, that our channel is functional. And what else? So we can see that the pore is wider than in the closed state, but is the channel open? So we cannot say this for sure, but we can do some molecular dynamic simulations to see whether the water and ions, they can actually pass through the ion channel pore. And they do. So you can see the water, it can pass through the channel. And for the closed state, wild type state, it does not. And also you can see that the ion, they also can pass through the ion channel pore. So again, this is independent evidence that indeed what we got, it was indeed the open state, the active state of the channel. So, and this is what we've done. So we solved the structure of TRPM7 channel in closed state, in nanodisk and detergent. We also solved the structure of the channel with mutation in detergent and without mutation, but in complex with the agonist. And we also studied several inhibitors of TRPM7 channel, and we obtained the inhibitor-bound closed states. And all these structures, they gave us an opportunity to propose the model how the channel works. So we see at least 4 different states for this channel. One, we have like resting closed state when the channel doesn't have any stimulus to be open. And then we see 2 open states, and they are different. Although they both open, they have channel pore open to conduct ions, but they have differences in this lower part, cytosolic part. So with the mutation, nothing happens in the bottom part of the channel. But when we add agonist, we can see a lot of movements in the lower part, intracellular part of the channel. So we propose that that's maybe the two independent mechanisms how the channel can be activated. We know from the literature that this channel is constitutively open. It means that sometimes it can be open just by itself. And I think that, and we think that that's what actually happens on the cell membrane. But when the cell has magnesium deficiency, it can force the channel to be open. So there are some, we speculate there are some endogenous ligands inside the cell that can force the channel to be open and conduct more magnesium. And if there are too much magnesium inside the cell, magnesium itself can block the channel, but cell can also recruit some additional compounds that can convert the channel into the closed state, inhibited state. And we can speculate, so this, how the channel can be regulated. Okay, so I want to wrap up. I want to acknowledge all my colleagues that helped me with this project, all the members from the Sobolevsky lab at Columbia University in New York, our colleagues and collaborators from the Carnegie Mellon University in Pittsburgh, who did MD studies, and also Vladimir Chubanov lab in University of Munich, who did most of functional stuff. And also want to thank cryo-EM facility of Columbia University. And also I want to thank you for your time. I will be happy to answer your questions.
So thank you very much, Kirill. So this was a really, really nice talk. And I thought that there are a lot of questions, but maybe I'm allowed to ask the first question. So you said you're using nanodiscs for your isolation. So did you try also the synthetic polymers or other synthetic polymers like SMAR?
That's so, yeah, this is an excellent question. So yeah, and you always, when you work with the membrane proteins and when you want to get the state that is like as closely as possible to be the native state, you also have to be very cautious about what detergent, nanodiscs you are using. So in our case, we kind of follow the standard rules of our labs, kind of standard protocols, but we always try to find better ways how we can extract our protein. And yes, this is now in our pipeline. We want to try synthetic polymers like SMALPs to try to stabilize our protein.
Okay, yeah, because so in the last weeks I was involved in launching, for example, the new sulfo-SMAR and sulfo-DIPMAR polymers, and they are, for example, or as they are described in literature, that they are quite good for cryo-electron microscopy. So especially for high magnesium or calcium concentrations. So maybe this is something for you. So we can send you, if you would like to have some samples from the polymer, so then you can try it by yourself. But our cryo-EM experts said they can be quite beneficial for the work and also for the solubilization and isolation efficiency. So
yes, so we will be happy to try them. Yes.
Okay. Great. So then let's come to the question and answer session. And the first question. So it was asked by Rowan Parker, and he's asking, are there any theories as to why cholesterol might interact with TRPM7?
Yes, so there are some literature that says that cholesterol activates TRPM7. So we think that because we always see cholesterol bound to our protein, and it doesn't go away when we do this extraction using, mild, but they still harsh detergents. So it should play some role. It should play some role. And again, literature says that it helped to activate the channel. We are doing some additional stuff. We're doing some mutagenesis around the cholesterol binding site. We want to kind of dig deeper in this topic. But for now, this is the only information that we have.
Okay, great. Thank you. So the next question is from Nikolai Rupinsky, and he's asking, can any of the order lipids you see explain the ability to bind the agonist with nanodisks?
Yeah, this is excellent questions, and unfortunately, I don't have a good answer to that. So what we think happens, again, this is just my theory. So what we think happens that when the protein is more happy in nanodisks, for some reason, probably because the nanodisks, it wraps around the TRPM7 channel, but it also leaves some room for regular lipids. So maybe this is crucial step for these activators. They are hydrophobic molecules. They need to have some membrane to bind first, and then kind of penetrate inside the cell membrane to find the binding spot. So that's my theory. But again, this is all just speculations for now.
So would it be helpful for you to just influence the lipids involved in the nanodisk formation? So, for example, by using cell-free expression systems and adding specific lipids for checking the binding?
So actually, yeah, this is a good question, but I don't think that it's actually possible to use cell-free expression to express such huge proteins. And not only you need to express TRPM7, these big channels, so the length of the channel is more than 1,000 amino acids, but you also have to assemble it somehow. So I think it's going to be tricky for cell-free expression, but when we do the nanodisk reconstitution, we can choose the lipids that we can add to form these nanodisks. And we actually do this. So we have some additional studies when we add different types of lipids to see whether these shift the equilibrium of like close to open state to like more open state, or maybe it can convert the channel into some new conformation.
Okay, nice. Great. So the next question was asked by Bernd Lünemann, and he's asking, is the improvement in resolution calculated with similar number of particles, and are the B factors comparable?
Oh, so this is excellent question. And yes, so the number of particles is very similar. So, and B factors for the TRPM7 in nanodisk are lower. So it means that the structure is like more stable, if you can say that. So yeah, but again, the B factors were also comparable. Yeah. So what we think happens that the nanodisks forms just kind of better environment for the protein than detergent, but still detergent does pretty good job as well.
Okay, thank you. So the next question was asked by Skatsai Yuri, and he's asking, are the structures in the closed state different in detergent and nanodisks?
No, they're the same, just the quality. So the structures, the conformation is the same. What we see a little bit of changes in the surrounding lipids, but even we don't even can quantify them. We can see a little bit difference, maybe like more detergent molecules that we see in detergent structures. But again, the structure and the map quality of protein is absolutely the same, like 100% same.
Okay, nice. So this is quite important information, right? So that it's not quite different.
Yep.
Great. So Ethan Henderson is asking, I'm also working with membrane proteins. May I ask which tag you choose for affinity chromatography and why?
Yeah, so the first question, so the first answer is kind of, I want to answer the second question. So we use whatever we have in our lab. And it looks like it doesn't make any difference which tag you use, but we use strep tag. Why? Again, this is just, we have this tag. It works very good in our case. And I saw many papers that used HA tag, flag tag, other tags, histidine tags, and they also work fine. So this is just kind of what we do in our lab. But you can choose whatever tag you want. I don't think this make any difference. Just whatever protocol is better established in your lab. You should go with this.
Yeah, I think you can, or you have to be maybe careful if you're using, for example, synthetic polymers. So if I remember correctly, there are some difference, but then I have to ask our protein purification specialist in the lab. So, but I think he told me once that there are some difference with respect to the synthetic polymers, but
Yep.
you have to be aware of it. Right. So the next question is asked by Carmen Castillo, and she's asking, why do you think the resolution of the cryo-EM images in Nanodisk was better than the run from the detergent micelles?
So I have like two explanations for that. So the first one is that the protein, at least I believe, maybe that is not true, but I believe that the protein in nanodisk is more stable than in detergent. And this gives you some bump in improvement of resolution. This is like one reason. And the second reason, I think it is more important, that when you use detergent and do cryo-EM in detergent, you have also some empty micelles, and they can add some background to your cryo-EM images and make them more noisy. So like in general, when in detergent, usually the quality of micrographs are worse. But when you use nanodisks, basically what you do, you convert your channel that was transmembrane channel with the hydrophobic transmembrane domain, you convert this channel into soluble particles. So basically what you do, you solve the structures of soluble protein with minimal background. And if you have minimal background, the noise is less, you have usually better quality of your reconstructions and higher resolution, better resolution.
Thank you. So the next question is from it's asked by Thomas Heger. So, and he's asking, do you have any explanation for the irregularity in the micelle shape visible in one of your detergent structures? And do sugars from detergent molecules interact with soluble part of your protein? Thank you.
Yeah, so again, this is like very good observation. I would rather not pay much attention to the quality of the micelle around the protein, because those are kind of, so I did these figures just to visualize the shape of the micelle. And if I tweak some parameters in this visualizing software like Chimera, I can do micelle shapes like more or less the same. So I don't want to pay like much attention to the visibility of the micelles. But the second question is about the sugars. Yes, indeed, we see sugars that are attached to our detergent. So GDN has kind of two arms with double I don't remember the name of the sugar, so we have kind of double sugar and like two arms. So 4 sugars are attached to detergent molecules. And we don't see whether they interact with the soluble part of the protein, but we do see that they are very close. That's what we can say for sure.
Great. So time is running, so let's come to the last question. So, but you don't have to be afraid. So every unanswered question will be answered by mail later. So the last question is asked by Victor Venturini, and he's asking, in order to generate your nanodiscs with MSP, you had to use detergents previously, which were subsequently depleted with the biobeads. Do you think that due to the use of detergents, although transitory, it could not be considered a native structure as the lipidic environment might have changed?
Oh, yes, this is absolutely true. So this is absolutely true. So although some people claim that nanodisk provides native environment for the membrane protein, this is not true like at all. So this